The topic of this presentation is on nanomedicine. We're going to be going over an overview of the different types of materials that are utilized in nanomedicine applications. And then we will be talking about what are some of the reasons why nanoscaled carriers are utilized a lot in this area, and then finally wrap up with some examples of various type of nanomedicines that are either in research or in the clinical setting. So to begin with, here are some of the reading materials that I suggest. The first set is from the Biomaterial Science book. There's a whole section on injected nanocarriers. Then we have a couple of review papers. The first one talks about the tumor microenvironment and basically providing some of the reasons why nanoscale carriers are utilized for targeted drug delivery to cancers. And also there's a review that actually has two parts on the nanomedicine revolution that talks about, again, the various type of nanomaterials that are being utilized, some of the advances in terms of nanomedicines actually going through clinical trials and into the market. And it also talks about some of the challenges associated with the field. And then we have two other reviews, one of them on the use of nanocarriers for targeted delivery to cancers. And then finally, one that is in general in terms of nanomaterials in biomedical applications, whether they're inorganic or organic polymer-like. So nanomedicines consist of injectable colloidal suspensions for the most part, where the nanoscale particles are designed for drug delivery, therapeutics, biosensing, bioimaging, and theranostic applications. Here I want to pause and talk about two things. Notice that I have mentioned injectable colloidal suspensions. What this means is that most of the times for most of these applications, what we're going to do is that we're going to prepare individual particles that are in the nanoscale, and in order to be able to administer them to the patient, those particles would typically be suspended in an aqueous environment in a solution like phosphate buffer saline, for example, which is basically an aqueous solution that contains the right ionic concentration to be of the same osmolarity as physiological fluids, so that it is biocompatible, basically. So in most of the applications, we're dealing with colloidal suspensions. Of course, we can also utilize nanomedicines in other contexts. For example, in biosensors or lab-on-a-chip systems, you might utilize particles that are part of a topography of another material, like for example, a nanocomposite, in which case a colloidal suspension might not be of relevance. But here we're really focusing mostly on injectable colloidal suspensions of those particles. Then in terms of the applications, here I have this concept of theranostics. So this is a word that has been basically created by merging the word therapeutic with diagnostic. So a theranostic nanomedicine would be one that could be utilized for both the detection and the treatment of a disease. For detection, for example, the particle could act as a contrast agent to be able to find the location of the diseased cells. And then from the therapeutic aspect, that same nanoparticle could act as a therapeutic agent by enabling localized drug delivery or maybe laser triggered photothermal therapy or something like that. Again, we're combining both capabilities in one single system. Nanomedicines for biomedical applications typically range from 5 or 10 to several hundreds of nanometers in size. And so this might be something that is different from the terminology that is used in other material science fields. So here it's okay to go to hundreds of nanometers and still call it nanotechnology or nanomedicine. Typically the lower end range is going to be about 10 nanometers. And the reason for that is that under 10 nanometers, particles tend to be rapidly cleared from the circulation through renal filtration, because basically they would behave as if they were small molecules below that. But if they are too large, obviously we're going to have problems with first being able to inject them, but also if they're large enough, they can be filtered out through basically they can be captured, I should say, by certain organs of the macrophage phagocytic system in our bodies. So typically we're talking about size range from about 10 nanometers to about 100 or maybe 200 nanometers tops. All right, in terms of nanomedicines and the scale of nanomedicines to biomedical applications, we can see here a scale. Obviously, we're talking about very small size ranges, but nanomedicines, you can compare their size to something like a virus in the order of 100 nanometers. Of course, either on the size range or bigger than your typical protein. So an antibody, for example, which is a fairly large protein, it's about 10 nanometers. And so we're talking in that range from the level of proteins to the level of to the size range of viruses or such. Of course, this size range is smaller than most of the cells that are in our bodies, which are going to be in the order of micrometers, something like 10, 20, 50 micrometers in size. And so the nanoparticles are relevant for biomedical applications because they're small enough that they can be injected. They're small enough that they can interact with the cells in our body at the cellular and molecular level, and they are large enough that we can actually functionalize them on their surface to include things like antibodies for targeting or other molecules that enable better interaction with the target tissues. And they're also large enough that they enable us to use them as carriers for therapeutic agents or imaging agents. So the size is very important. Also, obviously, they're going to have high surface area. And that's something that might or might not be useful in our application. Well, one of the reasons why it might be good is that having a high surface area again enables us to modify the surfaces in such a way that we can create a multifunctional nanoparticle that might have, again, a targeting agent or a fluorophore to enable us to track the nanoparticle. But on the other hand, having a high surface area might be problematic for our application because it also enables rapid absorption of proteins onto the surface, and it also can lead to aggregation. And of course, remember that we're talking about utilizing them as a colloidal suspension. So aggregation is not a good thing in our case. In terms of the market for nanomedicines, again, my data is kind of dated, but the market was expected to be about 1 trillion by 2015. So it's a large, large market. And most importantly, there's a lot of interest in the area. The National Science Foundation predicted that by our year right now, that 1/3 of the patents and 1/3 of the startups would be associated with biomedical nanotechnology. So basically a lot of activity in this field. They also predicted that half or nearly half of all future pharmaceutical products would have a nanotechnology component, whether that was a nanocarrier that acted as a drug delivery system, or maybe a targeting agent that was utilizing some kind of nanoscale biological molecule or something like that. There's a number of different types of nanocarriers, and here I just have pictures of some of them. We could have nanoparticles, which could be of very different types, but those nanoparticles are usually in the size range of 50 to tops 200 nanometers. As I mentioned, usually we actually want to keep under 100 nanometers if at all possible. Those nanoparticles can be made out of different materials. So we will be talking about gold nanoparticles, iron oxide nanoparticles, quantum dots, which are made from semiconducting materials. We can also have polymeric nanoparticles, of course, and those polymeric nanoparticles can be bare polymeric nanoparticles as shown here, or they can be surface functionalized nanoparticles that contain things like polyethylene glycol to enable better stability as a colloidal suspension, and also enable us to include ligands for targeting. Those ligands would be biomolecules that have high affinity towards certain molecules or certain receptors that are found on the cells of the disease that we're trying to target. Other than nanoparticles, we can also have micelles. So a micelle is actually a dynamic structure That's something that is important. So it's not a solid nanoparticle. As you know, my cells are made from the self-assembly of either block copolymers or amphiphilic molecules, if you would. But they are dynamic in that it is a reversible process going from the self-assembled structure to the solution of those individual molecules. And that process is controlled by a number of factors, including the concentration of the polymers, the ionic strength of the medium, the pH of the medium, the stirring, the temperature, all kinds of situations can reverse that process. And so this is something that can be useful for the case of drug delivery if you're able to maintain stability until the micelle reaches the location where you want the drug to be released, and then have the micelle disassemble. But it could also be a problem if your micelle is not stable and it ends up being disrupted before it reaches the target site. Micelles tend to be smaller than nanoparticles. They tend to be in the order of maybe less than 50 nanometers. After that, we have liposomes. Liposomes are made from, or they are spherical vesicles that are made from phospholipid bilayers, and that are where the phospholipid bilayer is basically surrounding an aqueous core at the very center. That aqueous core can be utilized to load hydrophilic drugs, and then you can also use the bilayer itself that contains lipids to be able to load a hydrophobic molecule. So they can be multifunctional in that they can load both hydrophilic and hydrophobic molecules. We're going to have a section on liposomes, so I'll leave it at that, but their size tends to be kind of in the same range as micelles and nanoparticles, from about 40 nanometers to about 400 nanometers. After that, we have, let's see, dendrimers. So dendrimers are highly branched macromolecules that have a controlled, very controlled three-dimensional architecture or three-dimensional structure, as you can see here. Actually, you cannot see very well. And so if you Google the word dendrimer, you can see more close up the chemical structure. And you can see that basically what we have is that there's a central core, which is a central molecule that is multifunctional. And then from that core molecule, we're going to have growth of several layers of other molecules that are attached covalently, kind of in a row through a chemical reaction. And so it's a very interesting architecture. We have a lot of functionality because basically you have N groups that are functional on every single part of the molecule. A problem with these dendrimers is that first, they're synthetically challenging to be made. And second, they tend to have significant cytotoxicity, and so applications of these in real life medicine has not been achieved. Then we have polymer drug conjugates. These conjugates are basically polymeric macromolecules that are made by taking a polymer and covalently functionalizing it with the drug that you're trying to deliver. And it could be, as shown here on the left, where you have one polymer that is attached to one molecule. Let's say that that triangle represents your drug. Or it could be a polymer that has several functional groups onto which you functionalize several molecules of the drug. And then that polymer conjugate is able to self-assemble according to the hydrophobicity of the drug. So in this particular case, the drug is hydrophobic, and so it wants to be away from water, and that's why you see that the drugs aggregate, and you have kind of like a particle that forms from that conjugate. A lot of times in these drug conjugates, the polymer of choice is going to be polyethylene glycol, because most of the times what you're trying to do is to increase the solubility of the drug that you're trying to deliver. If the drug is hydrophobic, it's not soluble in water, and so you conjugate it to polyethylene glycol to increase the solubility of that polymer, of that drug, I should say. So most of the times it is polyethylene glycol. Let's see, and from there we can go to solid lipid nanoparticles. As the name implies, they basically utilize lipids to be able to form a core that is hydrophobic. That particle would have to be stabilized in order to provide better interaction with the hydrophilic environment around it. And so here it's kind of being shown that is surrounded by what do you call it? Goodness. I just went blank. by phospholipids or some kind of amphiphilic molecule that would have a hydrophilic charged group, perhaps, to enable that particle to interact with water. Here on the right hand side, we have drug nanocrystals. A drug nanocrystal would be basically if you take a drug that is not water soluble and then you do some kind of mechanical stimulation in order to decrease the particle size of that drug crystal all the way to the nanoscale. For example, think about taking a drug and then putting it in water where it would be a solid if it's not water soluble, and then subjecting it to high shear stresses or sonication or something like that in order to decrease the particle size to the nanoscale. So basically the particle itself is completely made out of the drug molecules. And then polymersomes is basically very similar to the micelles or the liposomes, but they are made from polymers that self-assemble into a kind of bilayer as shown here. So for example, this could be made by utilizing a third block copolymer, meaning that you have a polymer that has three different blocks: hydrophilic for blue, red hydrophobic, and then another hydrophilic group. And it basically self-assembles into a form that is very similar to the liposome, where you end up with a core that is hydrophilic that would enable you to load a hydrophilic drug. Then you have a layer that is hydrophobic, where you could load a hydrophobic drug, and then the whole particle is stabilized by the hydrophilic part of the second hydrophilic part of the block copolymer that enables this to now be suspended in water. There are other types of nanocarriers, including nanoclays, silver nanoparticles, and polyplexes. Nanoclays, if you are interested, one of our faculty, Dr. Gary Beal, works in this field, so you are more than welcome to talk to him about it. But basically, those nanoclays are materials that are derived from natural sources, clays basically, and these tend to be one-dimensional materials in that they are, or two-dimensional, I guess, in that they are sheet type materials. Basically, we're going to have a very thin material that is maybe 1 or to a few nanometers in thickness, but they can be large on the other two dimensions, maybe in the micron scale. Silver nanoparticles are utilized for antibacterial properties, and I believe that we have a section on those. And then polyplexes are going to be complexes that are made by taking polyelectrolytes and putting them together. And one of the main applications of these polyplexes is to generate drug delivery systems for oligonucleotides, for either gene delivery or siRNA delivery or something like that. Obviously, the oligonucleotides have a negative charge, and so by utilizing a polycationic polymer, we can complex them to form a polyplex, and the role of the polyplex would be to stabilize the nucleic acid so that it is not as easily degraded by nucleases that might be present in the physiological fluid. All right, so from here, we're going to start talking about liposomes. So liposomes are ordered, self-assembled spherical nanostructures that are consisting of bilayers of phospholipids. Here on the right-hand side, we have a phospholipid molecule. That phospholipid molecule is typically represented in this graphical way, where the sphere represents a water-soluble group. Typically, it's going to be a charged phospho group, basically a phosphate that has a negative charge. And then this part is going to be an organic molecule, typically a hydrocarbon, or two chains of hydrocarbons, I should say. So this part is water insoluble. This part right here enables that whole molecule to interact with water. And so when you put them into water, they would typically self-assemble in this way. Now, I'll go ahead and make a side note. So remember that micelles are similarly made from amphiphilic molecules that have one side that is hydrophilic and one side that is hydrophobic. The micelles, however, tend to form that kind of spherical shape immediately, right? While the phospholipids tend to make layers by layers that are not spherical necessarily at the beginning. And the reason for that is that we have two of these hydrophobic molecules. So it is not as easily for this thing to self-assemble into a small micelle type structure, because you would need this part to be less bulky compared to the hydrophilic part that would be on the outside of the particle. So that's something to keep in mind. All right, anyways, when you put this into water, it forms a phospholipid bilayer. And that phospholipid bilayer can be manipulated then into forming a spherical type of particle, where the phospholipid bilayer is forming this shell, and the inside is actually going to be empty, or not empty necessarily. It's not going to be empty. It's going to have water. It's going to have an aqueous environment inside, which, as I mentioned, would enable you to encapsulate a hydrophilic molecule in this inner region. And you could also load a hydrophobic molecule within the very organic and very hydrophobic bilayer right here, where you have basically the tails of that phospholipid together. So that's what it was here. They can be easily modified chemically or physically, and as we're going to see on the next few slides, basically by selecting the right type of lipid molecule or phospholipid, you could have a phospholipid that would have functional groups that would enable you to attach other things, or you could actually use a phospholipid that already contains polyethylene glycol and then has a functional group on the very end. And you can blend a number of different phospholipids to make one single formulation of liposomes. So that's what we're going to see now. So here are the various types of phospholipids that can be utilized. This is phosphatidylcholine, cholesterol. I'm not even going to try to say this one. We call it DSPE, DOPE, and pegylated DSPE. This DSPE is the one that I have seen the most in the literature. And notice that we have two versions of it. And before I say that, actually, notice that all of them are going to have a hydrophobic, typically hydrocarbon-like chains for the hydrophobic part, except for cholesterol, which obviously most of the molecule is itself a lipid. It only really has one hydrophilic part, which is right here. But also all of the others, not only do they have those hydrocarbon chains, but they also have the hydrophilic portion, which is that phosphate group, which is negatively charged. And you might have a hard time seeing that negative charge, but trust me, it's right there in one of the oxygens of every single one of these molecules. So again, hydrophobic, hydrophilic, hydrophobic, hydrophilic. Now, looking at the two DSPEs, one of them is not pegylated, yet it has a primary amine group that you could utilize to include a targeting agent, or maybe to modify the liposome after the liposome has been fabricated. But similarly, you could utilize pegylated DSP that already contains a polyethylene glycol chain. And so when you mix these two together, you would end up with a liposome that would already be pegylated in some of the regions. So that's something that is important. And as I mentioned, typically in liposome formulation, you would mix 1, 2, or 3 different lipids to get a system that has the right stability, because obviously these are dynamic structures as well. They're not solid, right? So you need to have sufficient stability for that liposome to not break apart until it reaches a membrane. And actually, that's something else that I wanted to mention. So if we come back here, this structure and the bilayer that they are made out of is actually very similar to the structure of a cell. The cell membranes are made from phospholipid bilayers just like this. And so the way that these liposomes tend to work in terms of drug delivery is that they would maybe circulate in the body, reach the location where they are supposed to go to, and once they get there, they can actually fuse into the membrane of a cell because of the compatibility in composition. They can basically fuse, and when they fuse, they would deliver their cargo into the cell. So that's something to keep in mind, that makes liposomes quite interesting. All right, so yes, you would have to mix a couple of these to get the right stability. Typically, the more hydrophobic components that you have, the more stable that liposome is going to be. So a lot of papers, you will see that they also include cholesterol to make that system more hydrophobic and more stable. In terms of the preparation of liposomes, this is a figure that I obtained from Sigma Aldrich that actually it's pretty good at explaining what happens. But basically what we have is that we start by taking the powdered lipids and putting them together, mixing however many lipids you're going to be utilizing. And if you're going to include a drug, this is where the drug would also be included. And you put them all together, typically in a solvent. That solvent, or the most commonly used solvent, is going to be chloroform, CHCl3, or a mixture of chloroform with alcohols, things like methanol, for example. And so here you would dissolve the lipids all together. And the whole point of this initial solution is to be able to mix all of the lipids and all of the drug all the way to the molecular level to form a homogeneous system. After that, the solvent would be evaporated away, and you would further freeze dry that solid lipid mixture. And the reason for that is just to get basically a cake of lipid that has a homogeneous composition now. Now we're going to take this, and what we're going to do is that we're going to hydrate it. And to do that, we're basically going to add an aqueous solution that includes the water-soluble drug, if you needed a water-soluble drug to be loaded inside of the core of the liposome. And that aqueous solution could be just water, or it could be a saline solution that would have the right osmolarity for physiological use. But basically, the role of that aqueous solution is to be able to hydrate the lipids, and so that's shown here on the right hand side, a little bit in depth. But basically, that process, you're going to be hydrating and getting the lipids to, I'm not going to say dissolve, but to suspend in the aqueous environment and start forming the micellar structures or liposomal structures. Let me come back to this image right here. So we're starting with the dry lipid film, the dry cake of the lipid. We add water, and what happens is that the lipids start getting hydrated. They start swelling because of the interaction of the phosphate groups with the water, and they kind of start coming apart. They're not going to come apart into individual liposomes right away. They're actually going to form these multilamellar vesicles, which are going to have different shapes, different sizes, different layers. You can think about them like onion rings, basically, where you have layers, and in between each of the layers, there's going to be water. From there, what we are going to do is that we're going to apply a mechanical input, which could be either sonication or extrusion, in order to get now these guys to become smaller. So extrusion is basically where you take a suspension of these multilamellar vesicles and you force them through a filter of defined pore size. And basically what you're doing by that is you're forcing them into being smaller. By doing this process, however, you still end up with perhaps a couple of layers in that liposome, but it's going to be of a size that is less than the pore size of the filter that you have used. Typically, using this process, you get liposomes that are in the order of 100 nanometers. Alternatively, you can use sonication or homogenization, where you can utilize different types of sonicators. So some of you in the lab actually got to use a bath sonicator. That's one of the ways that it can be done. We can also utilize probe sonicators, which one of you did for your project. And basically through that, you're applying sound waves, which generate significant it's kind of significant mechanical steering at the molecular level almost, and what you end up with is small unilamellar vesicles that are in the order of 10 to 50 nanometers. Now, both of these processes I should have mentioned, and actually all of these processes are done at a temperature that is higher than what's called the TC, which is the temperature at which these phospholipids go from being a liquid crystal system to being a liquid. So it's a transition temperature that enables these to behave more like liquid type lipids. You can think about it like from the polymer perspective, it would have been something like going through the glass transition temperature or through the melt temperature, where we have broken up the interaction that you initially had between the dry solid lipids, and now giving them more mobility for them to be able to assemble. They need to be flexible to be able to assemble into these kind of spherical structures. So anyways, once you have formed this, then you are left with the suspension of the vesicles. So that is the process right there. In order to control the size, there's a lot of different tricks that can be played, including the type of sonication or extrusion that is utilized. In some processes, they will actually do several cycles of freezing and thawing. There's just a lot of different tricks that can be played in order to control the size and the polydispersity of these liposomes. On this slide, I wanted to show you kind of the evolution of liposomes as to like the first type of liposomes, which are basically just the liposome itself that includes either a hydrophilic drug that is loaded at the core or a hydrophobic drug that is loaded within the lipid bilayer. After that, we have now liposomes that might include antibodies for targeting, and those antibodies can be either covalently attached directly to the phospholipid, or they can be incorporated by, let me find the right word that they had here, they can be incorporated by hydrophobically anchoring the antibody. What that means is that you would have a phospholipid that maybe has a polyethylene glycol and that has a functional group where you can attach the antibody. And instead of covalently attaching it after the liposome is formed, you would actually include the antibody during the formulation of the liposome so that you can hydrophobically anchor it to the liposome. From there, we go now to the next, I guess, generation of these liposomes, which are known as the long circulating liposomes. By long circulation means that we are now pegylating them or incorporating a polyethylene glycol surface coating such that we can increase their circulation time in the body. Remember that polyethylene glycol, it's a hydrophilic polymer that can hydrogen bond with a significant amount of water. And so what you end up forming is basically a shield that is hydrophilic that would prevent proteins from absorbing to that surface and thereby increasing the circulation time of those liposomes in the body. In this particular case, this F represents a protein or an opsonin protein that would be the type of protein that would adsorb onto the micelle, and the polyethylene glycol is there to prevent that. Now, other than polyethylene glycol, we can utilize other hydrophilic polymers, but PEG by far is the most widely used. From there, the next generation are still going to be long circulating liposomes, but now that are also functionalized with targeting agents such as antibodies in order to enable specific interaction with particular receptors or biomolecules that are present at the disease site. And finally, the last generation are multifunctional liposomes that incorporate a number of different things. For example, we can incorporate drugs, we can incorporate imaging agents, we can incorporate magnetic particles for magnetic targeting, we can incorporate gold or silver particles for either electron microscopy or to provide temperature control. We can, of course, we're still going to incorporate surface functionalization such as polyethylene glycol to increase the circulation of the liposomes in the body. We can incorporate fluorescent molecules, which I think is this one, to enable tracking of that liposome and its distribution in vivo and within the cells. We can incorporate cationic lipids that enable us to complex oligonucleotides to the surface. There's just a lot of different things that we can incorporate, basically, is what I'm trying to say. Oh, and I wanted to mention one thing, and that's that this plot came from a review paper by Dr. Vladimir Torchilin. He is, I think, one of the main authors in the area of liposomes, and he's at Northwestern University, and it's just a good name of somebody in the field that you should know. All right, so there have actually been several formulations of liposomes that have been approved by the FDA. Some of them are listed here. The only one that I really wanted to talk about was Doxil. So Doxil is a liposome that is utilized for cancer therapy. It loads the drug doxorubicin within that liposome, and liposome is actually an amphiphilic drug in that it can be dissolved in aqueous environment, and it can also be dissolved in a hydrophobic environment. And in this particular case, it is being loaded in the core of the liposome. I'm not going to go into detail, but the way that they load it is actually utilizing some gradients of ionic strength in order to get very high concentrations of that drug in the core of this liposome. Now, Doxil is, if I'm not wrong, Doxil is actually a pegylated, yes it is. It is a pegylated liposome, meaning that we have polyethylene glycol on the surface. And we can see that here. This methoxypolyethylene glycol means that we have polyethylene glycol that is terminated in a methoxy group. Methoxy is where your peg now ends in, sorry, come on. Sorry, it's not writing. Ends in a CH3 group, OCH3 group. That's what methoxy is, which is exactly what I have shown here. So the end group is this methoxy group, which is quite stable. It cannot really be utilized for targeting agent attachment or anything. It's just a stable end group. And notice that in order to incorporate that polyethylene glycol, they utilize the DSP that has the methoxypolyethylene glycol. So it's not that the PEG is just on the surface, or well, I should not say that. It's not just that the PEG is attached to the surface. It's actually that it was anchored by utilizing a mixture of DSP with DSP that was pegylated, basically. All right, so now we move to novel metal nanomedicines. So in noble metals, basically, we're going to be talking about gold particles. They are typically regarded as non-toxic. There's a long history of their use in biomedical applications. The most commonly utilized are going to be gold nanomaterials and silver nanomaterials. And here on the right hand side, I show some of the images of gold nanoparticles of different diameter. You can see that according to the diameter, the color of that colloidal suspension is actually going to change. And here on the right hand side, I'm just showing what would be a single gold particle, just to show you that these particles have to be stabilized in the aqueous environment by utilizing stabilizing molecules. In this particular case, citrate, which is a charged molecule, negatively charged, and which also has significant content of hydroxyl groups that enables those particles to stay suspended in suspension. Now, the way that these particles are prepared is by reducing gold or silver reagents or precursors. For example, for gold nanoparticles, you're going to be utilizing chloroauric acid, or HAUCl4, in the presence of sodium citrate. And for the case of silver nanoparticles, you would be utilizing silver nitrate with sodium borohydrate as the stabilizer. The reducing agent also serves as a passivating ligand, coating the nanoparticle surface. So here the reducing agent is going to be the sodium citrate or the borohydrate as well. All right, so in terms of gold nanomedicines, there's quite a number of different architectures of gold nanostructures that have been utilized. For example, we can have nanospheres that are simply solid gold particles. Those are going to be typically very small in size, 1 to 100 nanometers, maybe on the large range. And those are shown here on the left-hand side. Nanorods, on the other hand, are going to be structures that have a different aspect ratio on two directions. The structure and the size of that structure is actually going to have some effect on the optical properties of that gold nanomaterial. For example, these kind of gold nanorods tend to have two peaks for surface plasmon resonance that can be utilized for basically irradiating those gold nanorods with two different laser lights to be able to provide effects such as photothermal effect. Then we have gold nanoshells. These gold nanoshells are typically made by taking a silica core and then growing gold on the surface of them. And then we have nano cages, which, there's different architectures as well. One thing that I wanted to mention again is that all gold nanostructures are going to have surface enhanced Raman scattering capabilities. Basically, you have a cloud of electrons that are on the surface of those metal particles, and those electrons reside in the conduction band. Those electrons can actually deflect incoming light, producing a reflective surface. And the light, if the light is of the same frequency as the oscillation of those electrons, then the particles can basically end up absorbing that light, or at least showing an absorption peak. And what that means is that we can again irradiate these type of particles with laser light, for example, and that laser light might be absorbed and then being able to be utilized as a way to generate heat at a local in a local way. One other thing that I wanted to mention in this slide is that the particle size for solid nanospheres, again, is going to have an effect on the color of those particles. And that is because the surface plasmon resonance is going to be located in different peaks of the electromagnetic spectrum, or different wavelengths of the electromagnetic spectrum. Two relevant companies that I wanted to point out are Nanospectra Biosciences and Nanohybrids. Nanospectra Biosciences works on the use of nanoshells for applications in photothermal therapy for cancer. They are actually, or they have been, and they continue to be in clinical trials for different type of cancer treatments utilizing photothermal therapy. In photothermal therapy, the particle would be irradiated with a laser, generate heat locally, and ideally be able to kill the cancer cells by hyperthermia, where now you would be basically denaturing some of the proteins in the cells and inducing sufficient stress to kill the cancer cells. Nanospectra Biosciences came out of the work of two or three female researchers at Rice University. Right now I can remember two of the names, who are Naomi Halas and Jennifer West. Jennifer West is no longer at Rice University. Last time I checked, she was at Duke University now, but the company remains in Houston. And then Nanohybrids is a company that works on utilizing gold nanostructures initially for photoacoustic imaging, where basically the nanoparticles would be irradiated with a laser. And when the laser light is absorbed by the particle would generate heat. But now the heat is utilized because of the vibrations that it would generate. That those vibrations could now be imaged by utilizing a sensitive ultrasound system. Basically, you have a pressure wave that develops, and that pressure wave can be detected using ultrasound. So initially, they were working on photoacoustic imaging, but right now I looked at the website just yesterday, and it looks like they're also starting to focus on the use of these gold nanostructures for other applications, including for biosensing, like lateral flow systems, for example. Nanohybrids came out of the work of two people at UT Austin, Dr. Stas Emelianov and Dr. Kimberly Homan, and actually also I forgot one more person, Dr. Konstantin Sokolov. They are actually some of my professors and one of my colleagues, so I'm very interested in the status of this company for sure. All right, so, and I should say Nanohybrids, it's in Austin. All right, so noble metal nanoparticles have a number of applications. We already mentioned some of the imaging applications, but for the particular case of silver, silver particles have antibacterial properties. And the way that that works is that the silver ions that might be released from those particles, specifically silver 1 plus, can interfere with some enzymatic function of the cells and lead to cell of those bacteria. In terms of the imaging properties, basically, we already mentioned that these particles have surface plasmon resonance, and that can be utilized for photoacoustic imaging, for optical imaging as well, and for photothermal therapies. One last thing that I wanted to show here is that for the case of gold nanoshells, you can actually tune the wavelength where that SPR peak is going to be found at by changing the ratio of the thickness of the coating to the diameter of the silica core. So if you have a thick coating of gold on the silica core, your SPR peak is going to be more blue shifted, while if you have a very thin coating, then you're going to be red shifting that SPR peak. And I mentioned that for gold nanorods, you can also tune that SPR peak by changing the aspect ratio of the length to the width of that nanorod.